All right, today we're diving into a virus that, honestly, is a source of constant fascination and maybe a little bit of a headache for any of us in clinical microbiology. We're talking about human herpes virus 6. It's a name we all know, but it's full of surprises. So let's jump right in.
And this is the heart of the matter, isn't it? On one hand, it's the culprit behind a pretty standard harmless childhood rash. No big deal. But on the other hand, this same virus can trigger fatal brain inflammation. How does that even work? That's the paradox we're going to explore. This incredible duality is what makes human herpes virus 6 such a clinical puzzle.
Okay, so to really get a handle on this, we've got to go back to square one. Let's get to know the pathogen itself. So, human herpes virus 6 belongs to the Roseola virus genus. But, and this is a big but, it's not just one thing. It's actually split into two totally distinct species, human herpes virus 6A and human herpes virus 6B. And this isn't just a minor difference. They target different doors to get into our cells. 6A goes for the CD46 receptor, which is pretty much everywhere. 6B, on the other hand, is a bit more specific. It targets the CD134 receptor, mainly on our activated T cells. And that one little difference in their choice of entry, well, that's the key to everything. It explains why they cause such wildly different problems. And here it is, the classic family photo for a herpes virus. It's got this beautiful, almost jewel-like icosahedral capsid. That's the 20-sided shell in the middle. Think of it like a tiny, super strong vault protecting the precious viral DNA inside. The whole thing is then wrapped up in a lipid envelope, which is studded with these little glycoproteins. Those are basically the keys the virus uses to jimmy the lock on our cells.
Let's zoom in a bit more. Right at the very center, you've got the core, the double-stranded DNA genome, the virus's blueprint. That's protected by the capsid we just talked about. Then you have this fascinating layer called the tegument, sandwiched between the capsid and the outer envelope. I like to think of it as a pre-packed toolkit, full of proteins and enzymes the virus needs to hit the ground running once it's inside a cell. And finally, that outer envelope, it's actually stolen from our own cells, a clever bit of camouflage that carries all the machinery the virus needs to infect the next cell. It's just a really slick piece of engineering.
All right, so we know what it looks like. Now let's talk about what it does. This is where things get really interesting, where we see that split personality in action.
Okay, so for the vast majority of people, your first meeting with human herpes virus 6B happens when you're a little kid. You get what we call exanthem sabutum, or as it's famously known in the Netherlands, Zesde Ziekte, the sixth disease. The playbook is always the same, right? A kid gets a really high fever for 3, maybe 5 days, and then, poof, the fever breaks. And right on cue, this non-itchy, pinkish rash shows up, mostly on the torso. It's harmless, it goes away on its own, and pretty much everyone gets it.
But now, let's flip the coin. Take that same virus, lying dormant in a transplant patient whose immune system is suppressed, and the story changes completely. It becomes a nightmare. Reactivation can lead to absolute devastation. We're talking about things like acute limbic encephalitis. That's inflammation of the brain that can kill you. It can shut down the brand new bone marrow, cause pneumonitis, hepatitis, the list goes on. It's the exact same virus, but the outcome? It couldn't be more different.
And this, of course, brings the problem straight to our doorstep in the lab. This is the big diagnostic dilemma. When we see this virus, how do we know if we're looking at a real active fire or just a little bit of smoke?
So our go-to weapon in this fight is, of course, quantitative PCR. Our qPCR assays are incredibly sensitive, they're specific, and they let us put a number on it to quantify the viral load. It really is the undisputed gold standard for tracking this virus.
But here's the million-dollar question. The qPCR result comes back and the viral load is through the roof. What do? Is this an active, life-threatening infection that needs immediate and, let's be honest, pretty toxic antiviral drugs? Or is it something else entirely? Because making the wrong call here, well, the consequences can be huge.
And that something else is a fascinating curveball called chromosomally integrated human herpes virus 6, or CIHHV-6. Get this, for about 1% of the population, the virus isn't just hiding out in a few cells. No, its entire genome is literally stitched into the chromosomes of every single nucleated cell in their body. It's inherited. It's part of their human DNA. And that means their blood will always show a high viral load whether the virus is active or not. So how on earth do you tell the difference between a real infection and this inherited artifact? Well, thankfully, there's a clever way. It all comes down to the sample type. In a true active infection, the virus is replicating like crazy and spitting out new particles into the bloodstream. So you'll find high viral loads in both whole blood and in the cell-free plasma. But with CHHV-6, the viral DNA is locked away inside the cells. So you'll see a super high load in whole blood, but the plasma, it'll be low or maybe even undetectable. That's why testing both samples in parallel isn't just a good idea, it's absolutely critical.
Okay, so that's the diagnosis. Now what about treatment? As you can probably guess by now, the approach to management is just as split as everything else about this virus.
This comparison really just says it all. Over on the left, you have the Dutch RIVM guidelines for a healthy kid with Zesde Ziekte. The advice, supportive care, maybe some paracetamol if they're uncomfortable. The whole goal is to reassure the parents and demedicalize a simple childhood illness. Now look at the right. These are the European ESIL guidelines for a transplant patient. The recommendation, aggressive intravenous antivirals like foscarnet or ganciclovir for weeks on end. Here, the goal is literally to prevent death. It's just two completely separate universes of medicine, all because of one virus.
So to wrap things up, let's talk about the challenges that are still out there. Because even with good guidelines, this is a virus that keeps us on our toes.
And just like we see with its cousin, cytomegalovirus, we have to worry about antiviral resistance. For ganciclovir, the virus can mutate its U69 protein kinase gene, which means the drug never gets activated. It's a dud. For foscarnet and cidofovir, we see mutations pop up in the U38 DNA polymerase gene, which basically changes the lock so the drug's key no longer fits. For patients needing long-term treatment, this is a huge, huge concern.
So that brings up the question of prevention. How do you prevent infection with a virus that's basically everywhere that most of us are already carrying? The short answer is, you don't. Infection prevention here isn't about masks and isolation. It's all about managing the host. It's about carefully managing immunosuppression in our vulnerable patients and being incredibly vigilant, always watching for the earliest signs of reactivation.
So what does the future hold? Well, there are a few big things on the wish list. First, we desperately need to standardize our qPCR testing across labs, internationally. We also need to nail down validated viral load thresholds that tell us exactly when to pull the trigger on treatment. And of course, we need newer, better, and less toxic drugs. But maybe the most exciting direction is looking at immunotherapies, like adoptive T-cell transfers, where we can give patients the specific immune cells they need to fight the virus themselves.
And that brings us full circle. Human herpesvirus 6 really is the ultimate virus of contrasts. Benign childhood illness on one side, devastating and deadly encephalitis on the other. It's this Jekyll and Hyde personality that makes it such a persistent clinical challenge. And, let's be honest, a completely fascinating puzzle for us in the lab. Just so you know, all the information we've gone through today was put together using a Gemini deep search. If you're interested in digging into the primary literature yourself, we've put a detailed list of all the sources in a Notebook LM document, and you can find the link for that down in the description.
Well, that's it for this deep dive. If you enjoyed this, please do subscribe for more explainers like it, and I would genuinely love to hear from you. For all the microbiologists watching, what are the biggest headaches and challenges you run into with human herpesvirus 6 in your own lab? Let us know down in the comments. I'm really keen to see what you have to say. Thanks so much for tuning in.